Bismillahirrahmanirrahim. We start with the A-level biology November 2020 question paper 1-1, and we'll try to handle up to 20 questions in this video. We all know in the 2021 syllabus this is only for 1 hour, but from 2022 this is going to go to 1 hour 15 minutes, which means that the MCQs will become a little more difficult than what they are now. And of course, this will mean that you need to be really clear about the concepts of biology. Start question 1. It says which row shows the correct order of size of these cell structures, and then of course it gave you a clue, largest to the smallest. Now if you look at it, what have they given you? Width of a mitochondrion, then width of a ribosome, then width of a cell surface membrane. Now if this is the cell membrane, the cell surface membrane, the width, not the length of it, but the width of it, which means it is only 7 nanometers. This is the width of the cell membrane, how thick it is or how thin it is. This is what is the width of the cell membrane. Now that would be the smallest. So width of the cell membrane was 3. So if 3, then what are these are the possibilities? This, and this. But then when you look at it, it says width of a mitochondrion, and you know mitochondrion, if I've drawn this to scale, this is a mitochondrion. And this is a chloroplast. So the chloroplast is larger. Of course, the largest organelle is the nucleus. So if I have to draw the nucleus, I would draw the nucleus about this big, because it is bigger than the mitochondrion and is bigger than the chloroplast. So what you have to understand is that the chloroplast would be the biggest, that is by 4, and then 1 would be the mitochondrion, and then 2 would be a ribosome. Ribosome is only about 60 to 70 nanometers, and this is 7 nanometers. So if I say this is the ribosome is this structure here, this gold structure here, which is about 70 nanometer. The rest are, of course, this is about 2 to 3 micrometer. This is about nearly 5 micrometer. And the nucleus is about 20 micrometer. So now revising all these different, so you need to go through a table which tells you the different sizes of the organelles and which one is the largest, and then of course we go down to the smallest. Now looking at question 2, the diagram shows a stage micrometer scale viewed through an eyepiece containing a graticule. The small divisions of the stage micrometer scale are 0.1 millimeter. So, I mean, somebody asked me, but miss, how do we find out which one is which? Well, of course, if you look at the later part of the question, it says the stage micrometer is replaced by a slide of a plant cell. So, I mean, the stage micrometer has been removed. So that is the one which was the stage micrometer was 0.1 millimeter. And that is the one which is always got some units to it. The eyepiece graticule has no units to it. Never, ever has it any units to it. So now 0.1 is equal to 40. So we do a simple calculation. We do the unitary method. 40 is equal to 0.1 millimeter. Then you convert millimeter into micrometer. So 0.1 into 1000 becomes micrometer, which becomes 40 is equal to 100 micrometer. So 1 is equal to 140. But then what did they ask you? They said, what is the actual length of the nucleus of the cell? So now if you look at this diagram, I'll just rub it off and then show it to you again. The nucleus is this part. Then you see how much is the nucleus, and you have roughly an estimate is about 10. So if 1 is equal to 100 over 40, so 10 is equal to 100 over 40 into 10, and that is 25 micrometer. You see, what you have to understand is, look at all the units here. All the units were micrometers. So you had to convert, of course, only this one is in millimeter, yes. But so micrometers was 3 were in micrometers and 1 was in millimeter. So then you can easily figure it out. If it's 25 micrometer, then how many millimeters is that? You see, 1 millimeter is equal to 1000 micrometers. So then you multiply this and it will become micrometers, but you knew the answer was 25 micrometers. So this is how you are going to figure this out, and this is how you do this question, is that you remember this, that what is given in the question is the stage. Now it says in the stage micrometer and the eyepiece graticule, and the stage micrometer scale is 0.1. So the one with these bold lines, which I'm just going to color in green, these bold lines and which is the unit. This is the stage micrometer. Insulin, question 3. Insulin is a hormone synthesized from two polypeptide chains inside cells of the pancreas. Which row shows the cell structure that are used in producing insulin and releasing it from the cells? So we know lysosome is not used in that. Lysosome contains hydrolytic enzyme. So lysosomes was not required because insulin would be made on the rough endoplasmic reticulum and then it would be shifted to the Golgi body where it would be modified and then it would be formed into a secretory vesicle and then it would be thrown out of the cell because it told you in the question synthesized and releasing it from the cell. So the releasing it from the cells would be by exocytosis. And of course, that is only possible if you know there is a cell and then it goes to the Golgi and then from the Golgi it buds off and then of course it comes here and then it just releases it out and then this is of course what you see is this sort of a structure left here and then this insulin is released out of the cell. So question 3, the answer was C, Golgi body you need and the rough endoplasmic you need. Then coming on to question 4, the electron micrograph shows a cell structure in a eukaryotic cell. Now if you remember this structure, it's got these three things. It's a very typical structure. Which statements about this cell structure are correct? ATP is synthesized in this cell structure. If it was the mitochondria, then that would have been the correct answer. The cell structure is made of protein molecules, and the cell structure replicates during interphase of the cell cycle. So during the G2 phase of the cell cycle, interphase of the cell cycle, the centrioles replicate. Replicate means we have one centriole. Centrioles are these rod-shaped structures like this. And then two are formed. So centrioles replicate. So that is why 2 and 3 was correct. And of course ATP is not right because ATP is not synthesized in the centrioles. It's synthesized in the mitochondria. So this was question 4. Now let's go on to question 5. Now when you look at question 5, it's a very interesting question. It's also come many times in a previous paper. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. Synthesis of polypeptides, synthesis of lipids. You know, synthesis of polypeptides is on the RER. Synthesis of lipids is SCR, smooth endoplasmic reticulum, and then packaging of hydrolytic enzymes that will remain in the cells. And that is, of course, the lysosomes. And lysosomes are Golgi vesicles. So the appearance were listed by letter. V membranes which surround an enclosed inner cavity. W non-membrane bound spherical structures. X a double membrane interspersed with pores. Now this of course is the nuclear membrane or the nuclear envelope. Both these words are double membrane interspersed with pores, not channel proteins, pores. Then non-membrane bound cylindrical structures. Then membrane bound sacs arranged as a flattened stack. Now that's always a very typical Golgi description. I always think of it as like a stack of puris, like you have halwa puri, and then a stack of these puris would be like the Golgi body. Now it says which student correctly matched the numbered function with the appearance of the cell structure. So now if you look at one was synthesis of polypeptides. Now synthesis of polypeptide is RER. Now which one is that? Is W non-membrane bound spherical structures. What are these? These are ribosomes. What are ribosomes made up of? Ribosome is not made up of the phospholipid bilayer. Please remember when we talk of a membrane, it's a phospholipid bilayer. Now phospholipid bilayer is present in the mitochondria, outer membrane, inner membrane. Then it is also present inside chloroplast. There is an outer membrane. Then there is an inner membrane, phospholipid bilayer, membrane present. And then even the thylakoid is made up of membranes. So thylakoid, so phospholipid bilayer, outer membrane, inner membrane, phospholipid bilayer, thylakoid membrane, phospholipid bilayer. In the mitochondria, outer membrane, phospholipid bilayer. So number one was W, synthesis of polypeptides. Two was synthesis of lipids. Now, what is synthesis of lipids? Is the SCR. Now, what is V? Membranes which surround an enclosed inner cavity. If you look at SCR, they look something like this. So they have a membranes which surround an enclosed inner cavity. Now that's a description which they give for smooth endoplasmic reticulum. And then the third one was packaging of hydrolytic enzyme. Now that of course is the Golgi body, and that was Z, which I've just talked about, is membrane bound sac arranged as flattened sacs. There's something like this. As I said, when you go to buy halwa puri and the puris are stacked against each other in the top, they just keep on putting them one on top of each other, and that's what it reminds me of, the Golgi body. And then of course vesicles will break off from them, and these vesicles are lysosomes. So packaging of hydrolytic enzyme that will remain in the cell. Lysosomes remain in the cell, they're not thrown out of the cell, they will remain in the cell to actually digest the worn out organelles. To question number 6. The DNA of typical prokaryotes is naked and circular. Naked means it doesn't have any histone proteins, and circular, means this is circular DNA. Doesn't have ends, it's just like a loop, like a rubber band. Which statement describes how the DNA, now this is where you did not read, DNA of eukaryotes differ from the DNA of typical prokaryotes. So only DNA of eukaryotes has proteins attached to it and is linear. So why are the other ones wrong? Only DNA of eukaryotes has a nuclear envelope. They are not telling you where the DNA is. They're not asking you. They're saying, how is the DNA different? And you're saying it has a nuclear envelope. Well, that's not what's in the question. The next question says, which describes how the DNA is different. Now I'm saying how the chair in my room is different from the chair in the dining room. Now you don't have to tell me the dining room. I'm asking you how the chair is different, and you'll say, well, the chair that has got a cloth on it, this one does not have a cloth on it, it has sort of a net on it or some jali on it. So you have to explain to me how the DNA is different. You don't tell me, okay, the DNA is present inside the nucleus and the nucleus is bounded by a nuclear envelope. So, that is wrong. Then only DNA of eukaryotes has a nuclear envelope around it. DNA, this is the nucleus. The chromosome is inside it, we say made up of DNA. So they're asking us how this DNA is different. How this DNA is different, not where it is present. Only DNA of eukaryotes has proteins. Only DNA of eukaryotes has proteins attached to it and is a double helix. Now you're not telling me how it is different. So, and is a double helix, but the DNA of prokaryotes is also a double helix. Double helix are both. DNA of prokaryotes and eukaryotes is a double helix. A student carried out the Benedict's test on two different types of milk, X and Y. A sample of each type of milk was heated to 100 degrees centigrade in a water bath with Benedict solution. The time taken for the first appearance of a color change was recorded. The results are shown in the table. Type of milk XY, time for the first appearance 13 seconds, second appearance 26 seconds. So one took lesser time and the other took longer time. Now, which row shows the biological molecule the student detected in each sample of milk and the sample of milk with the highest concentration of this biological molecule? So, biological present glucose, glucose, reducing sugar, reducing sugar. Sample of milk with the highest concentration was either XY or XY. So, you remember the first appearance of the more the glucose, the more it will react with the copper sulfate and the quicker it will change color. So 13, the one which took 13 seconds would have more. So highest concentration, that's why would be X, and it would be a reducing sugar. We do not test for glucose. We just know Benedict's is a reducing sugar test, and there are many, many, many different reducing sugars. So could be anything. Glucose could be some other reducing sugar as well. So reducing sugar, but it's not, Benedict's is not a test for glucose. It is for reducing sugars, and glucose is one of the many reducing sugars which we study in biology, right? So next question. Now when you look at the next question, Now that is question number 8. The diagram shows the structure of a monomer. Which molecules contain this monomer? Now you have to look at carbon 1. This is the rule that we follow. This is carbon 1. So if you look at carbon 1, let's look at this diagram here. So this is 2, this is 3, this is 4, this is 5, and this is 6. So on carbon 1, if you look at the diagram, the OH is below. So if the OH is below, so it's alpha glucose, it is below, beta glucose, it is above. And that's the mnemonic for it. Alpha below, beta above. Alpha glucose is below. The AOH is below on carbon 1. And on beta glucose, it is above. And beta glucose is only cellulose. So which molecules contain this monomer? So we have amylopectin, which is a part of starch, and is alpha glucose. Glycogen is also alpha glucose, and sucrose. So B would have, all the three would be present. So which molecules contain this monomer? So A was incorrect because just amylopectin and glycogen. C was incorrect because just amylopectin and sucrose. But B was correct because it was the three of them. It is present in all three of them. So this is what we had to understand about this. Now coming to question number 9, which molecules are formed by condensation? Condensation is the removal of hydrogen which involves a carboxyl group. Now this is of course collagen, beta globin, and triglyceride. And of course if you need to go through, please revise the chemistry of it and then you would know what do we mean by this. Then coming to question number 10, which row correctly matches the function with the structural features of cellulose, collagen, glycogen, or triglyceride? Now, of course, molecule, cellulose, triglyceride, function, support, energy source, and fibrous molecules held together by hydrogen bonds, and there are no branch chains. There's cellulose doesn't have branch chains, and triglyceride also doesn't have branch chains. So that was the correct answer to this. To question 11, this is a very difficult question in the sense that you have to spend a lot of time on it. It requires a lot of marking and everything which you have to do on the paper. You just can't do this theoretically in your mind. So the diagram shows the amino acids in a polypeptide, and they've given you type of amino acid, and they've given you these different shapes and the different shades of the dark black is polar basic, that the white hexagon is polar acidic, and then the gray hexagon is polar, and then the sort of the oval is non-polar. Now, when you look at this question, it says an enzyme catalyzes the hydrolysis of any peptide bond between a non-polar amino acid and any polar amino acid. Now, how would I do this question? How many small peptides and single amino acids will be formed? You see, how would I do this question is, first of all, I would look at all the non-polar ones. So I've given this a green color. So I would look at all the non-polar ones and I would sort of just shade them. And then I would start looking at, do they make a bond with any of the polar ones? And the polar ones are all these ones which are in red, this one, and this one. So you see, you have to really see, do they bond with any of these others? Because you see, if they bond with each other, like in this situation, all of them are together. Now, this is not where it will cut. These are all non-polar binding with each other. But then you see the non-polar when it binds with, now given you the red marks where it was cutting it. So if you count these, now let's do a count and see how many individual amino acids are there. Now the individual amino acids were very easy to figure out. Why? Because there are just a few of them. One, second, third, Now let's do this once again. So 1, this is the one which is an individual amino acid. Then the second one is here, second. Then the third one is here, and the fourth one is here. So we have 4 individual amino acids, and then we have 13 small polypeptide chains. Now I just want to number those. You can see I've just numbered them. So this is 1, this is the second one made of 2 amino acids, then 3, then 4, and then 5 is this one from here to here, and then 6 is this whole, this is up to here, sorry, and then 6 is this one from here to here, and then 7 is this one, and then 8 is this entire one up to here, and then 9 is these 2 amino acids, and then 10 is this one up to here, and then 11 is this one up to here, this whole thing up to here, 11, and then 12 is this one, and 13 is this one. So the answer to this was 13 and 4, which is the correct answer to this. So this was a little difficult. You need to figure this out. You need to do this 1 or 2 times, and I'm sure you all can get this done. And the question was, which properties of water reduce temperature changes inside cells: cohesion, latent heat of vaporization, and specific heat capacity. Now 1 and 2, 1 and 3, 2 and 3, and it was 3 only because specific heat capacity is the only thing which ensures that the temperature changes inside the cell are kept constant, and we are maintained at 37 degrees Celsius, or even plants or animals are maintained. Then coming to question 13, which enzyme is extracellular? Amylase in saliva is an enzyme that catalyzes the breakdown of starch in the mouth. That is an extracellular enzyme. Then coming on to question 14, the graph shows the concentration of components of an enzyme catalyzed reaction changes with time. Which line represents enzymes with active sites? But you know, more the active sites are empty at the beginning, and then they slowly decrease, and then of course they're empty because maybe all the substrate is finished out. Then 15, which effects can non-competitive inhibitors have on enzyme controlled reactions? They lower the Km value, they reduce the concentration of the product, reduce the rate of metabolic pathway. Now, naturally, when you have a non-competitive inhibitor, if you look at the graph of a non-competitive inhibitor, it is going to block somewhere, attach somewhere other than the active site and change the shape of the active site. So it will reduce the concentration of the product. So that is why the answer was 2 and 3, and reduce the rate of metabolic activity. So this is all what we had to do for question 15. Then question 16, which processes can allow transport into or out of a cell? Now, active transport, facilitated diffusion, osmosis, all can occur. Why? Because active transport is low to high. Now, whether it's low inside or low outside, it can occur. Facilitated diffusion is from a high to low. So if the inside could be high, then it could be going out. If the outside is high, it could be going the other way around. And osmosis is the movement of water molecules from a higher water potential to a lower water potential. So that could be any side, could be inside the cell or could be outside the cell. So all 3 were correct, which is A, 1, 2, and 3. Then we come on to question 17. Now, when you look at question 17, now that is a plant cell were put into one of the 3 different concentrations of sugar, 10%, 5%, and 2.5%. The cells were left for 50 minutes and then observed using a light microscope. Cell X, you saw what the vacuole is shrunk. Cell Y, it is turgid. Cell Z, again, the vacuole has shrunk and some sort of plasmolysis is occurring. Which statements are correct? Cell Y has a lower water potential than the sugar solution it was put into. Yes, cell Y, because it was less water potential, so water entered and become fully turgid. Cell wall prevents it from bursting. Cell Z was put into a 10% sugar. Yes, because it was the highest concentration, so water has moved out. Then cell Z has a less negative water potential than the sugar solution it was put into. And you must know what is less negative and what is more negative, and you must be very clear on that. Less negative means less solutes. Minus 10 is less negative. Minus 20 is more negative. Minus 10 means it has 10 solutes. Minus 20 means 20 solutes. If it is more negative, then of course it has more solute, so it has less free water molecules and less water potential. Then coming to question 18, during telophase, of mitosis, a scientist stains the chromosomes of a diploid animal cell with a fluorescent dye to observe the telomeres. You know, these are the chromosomes, and the telomeres are at the ends. These are the telomeres. We call these the egglets, which are found on the shoelaces. So during telophase of mitosis, a scientist stains the chromosomes of a diploid animal cell with a fluorescent dye to observe the telomeres. This cell has 22 chromosomes. Naturally, if you have 22 chromosomes, how many telomeres will the scientist observe in one of the nucleus? So 22 chromosomes will have 44. One chromosome has two. The telomeres are at the ends. So this is a telomere and this is a telomere. So two telomeres on one chromosome. So the answer was 44. Then coming to 19th, methotrexate is a drug used to treat cancer. It can act as an enzyme inhibitor, preventing the synthesis of a nucleotide containing thymine. Cells treated with methotrexate are not able to complete the cell cycle. A cell entering telophase is treated with methotrexate. Which stage of the cell cycle will be affected? Naturally, DNA replication only takes place in the S phase of interphase. There is no DNA replication in mitosis in the prophase, metaphase, anaphase, and telophase stages. So interphase is the part where DNA, so it is going to, it says, ends up preventing the synthesis of nucleotides containing thymine. And thymine is only used, nucleotides are only used in DNA replication. And that is the S phase of interphase. Going to question number 20. Which stage of mitosis is correctly described? In anaphase, chromosomes line up across the equator. In metaphase, in prophase, in telophase, chromosomes uncoil to form chromatin. So, in prophase, chromatids move to opposite poles. No, that's not very correct. Chromatids do not move to opposite poles. The centrioles move to the opposite poles. Then in metaphase, the centrosome replicates. In anaphase, chromosomes line up across the equator, right? Then coming to 21, the diagram represents a nucleotide containing cytosine. Which statement about this nucleotide are correct? This carbohydrate could be ribose. No, I'm sorry. It could be ribose. Yes, that is, of course, could be possible because it says it's a nucleotide. And nucleotides are those which are, could be DNA, could be RNA. So which statements about this nucleotide are correct? The carbohydrate could be ribose. Yes, could be deoxyribose. The organic base contains nitrogen. Yes, that's also correct. The base pairing occurs with guanine. Yes, cytosine always pairs with guanine. That is a base pairing rule. And the rule is that C is the third letter of the alphabet, so three hydrogen bonds form between cytosine and guanine. Always C forms three, and I always tell you to remember it that C is the third letter of the alphabet, A, B, C. Cytosine is a purine. No, I'm sorry. Purines are adenine and guanine. I told you to remember it. U for purine, AUG, adenine and guanine. So, purines. Question 22, the last question that we do here. A section of DNA contains 73 base pairs. Okay, we'll leave this for the second video. Okay, I complete this video here and we'll continue it into the second video from question 22 onwards. And thank you very much.